My name is Sarah, and I'm the managing director at CGEM, which basically means that I oversee a lot of the center's day-to-day operations, and I make sure that all of our projects stay on track. And so I trained as a structural biologist, but I've been working with CGEM away from the bench since 2019. And today I'll be talking about bespoke molecules from an ancient ribozyme, trying to give you sort of an overview of the research program of the center. So first, I want to give you a sense of who the CGEM team is. So CGEM is an NSF-funded Center for Chemical Innovation that started in 2017 as a pilot and got full funding in 2020. So these days, we're the fortunate recipients of $4 million in taxpayer funding per year from the National Science Foundation. So the center is headquartered at UC Berkeley, but we have faculty and students and postdocs at 5 other institutions across the United States. Everyone on the team brings different perspectives to CGEM, from synthetic chemistry to engineering, biology, and nanoscience. And this enables us to ask questions that no one lab could have asked by themselves. And in this talk, I hope I can convince you that CGEM's doing some really cool science and that there are plenty of opportunities for players to get involved. But first, I wanted to do a poll and see who might already be familiar with CGEM. So over the years, we've launched 3 different Eterna challenges, most recently the mini PTC and the Tbox challenges. And I think Jonathan is going to throw up a Zoom poll so that you can tell us which you have been a part of.
Give folks 60 seconds or so.
Great. Oh, cool. Okay, so it looks like we have a lot of people here who have participated in 1 or more CGEM challenges. Most people seem to have done the Tbox, but we have a good number of people who have helped us redesign the mini PTC or the P7A7. Mini PTC, as well as about 29% of responses said none of the above. So hopefully, if you haven't been part of CGEM before, you'll be excited to engage. And if you've participated in one challenge, you'll get a sense of how your work fits into the bigger picture. So I want to start by talking a little bit about polymers. So at CGEM, we think of polymers in sort of 2 categories, the first of which are chemical polymers. And so these are things like Kevlar, which is a copolymer made of these little aramid units. And we know Kevlar, it's lightweight, it's really strong. It was developed by this woman, Stephanie Pollack at DuPont in the 60s. We also think about materials like plastics or these nylons, which are made of polyamines. And we know nylon is really stretchy and flexible and present in a lot of sort of sportswear. And even polymers like those in your screens, which are made of these sort of repeating phenylene-violene units, are different polymers that exist in the world. And each of these chemical polymers are made of very different types of monomers. You can see by looking at the chemical structure that these look different from one another, and they're connected in different ways. And those connections between them give the products really unique properties. But when we look at polymers like that in sort of a bulk sense, there isn't a lot of control over the length. And they're usually the repeating unit of maybe 1 or 2 different monomers or beads on the string. And so there's a lot of different chemistry. We love the diversity in these chemical polymers, but you don't get a lot of control of your products. In contrast, biological polymers have highly defined sequences and lengths and really unique structures. And biological polymers include DNA, RNA, which you all know a ton about, as well as proteins, but those are really the only types of chemistries that exist in biological polymers. And so if we look at a protein here, it's an alpha peptide polymer, and you can see that the sort of backbone or the trunk of this little branch is all the same repeating pattern, but these side chains are where the different chemistries come into play. And so I've colored each of the side chains a different color, and in the language of proteins, this peptide says CGEM at Cal. And if we look at what a protein looks like in 3D, we know that its structure gives it really specific folds, and that these folds are really critical for its emergent functions like life. And so some proteins provide structure, like keratin in our hair or collagen in our skin, while others carry out the chemical reactions that keep us alive. And we'll talk a little bit more about this in a minute. But for now, I want you to think about the molecules that exist in the middle of these 2 things. What would happen if we could combine the chemical diversity of these chemical polymers with the sort of very defined sequence, length, and structure of biological polymers? And that's what CGEM is most interested in. So our grand challenge is to generate what we call sequence-defined chemical polymers, or these things in between that don't currently exist. So for example, what would happen if we could make a chemical polymer that had a defined sequence, like a material that contains a few units of Kevlar and a few units of nylon in a really particular order. Would it be super strong and flexible? Would it be something else entirely? What happens if you mess around with that sequence? And we're also, on the other side, interested in proteins that look a little bit different than normal. So maybe it's, an antibody that has an embedded natural product antibiotic right in the middle of it, so that it finds the cell it needs to find, delivers the antibiotic. Or maybe a few of the backbone connections in the protein look a little bit different than they do in nature, giving it some different properties. So I like to think of this quote from Walter Isaacson a few months ago. He said that molecules are becoming the new microchip. And I think it really emphasizes what CGEM wants to do. So Isaacson is referring to the revolutions in genome sequencing and gene editing that have made it possible for us to code for molecules in the same way that we code software. And in the background of the slide, you can see what he's talking about. So many of you are probably familiar with the central dogma, the idea that the instructions for a protein are transcribed from DNA, shown here in yellow, into messenger RNA in this sort of teal color, and that messenger RNA is translated by the ribosome to a chain of amino acids that fold into a protein. And these RNA molecules called tRNA really help connect the code embedded in the mRNA and help the ribosome put the amino acids in the correct order on the string. And so the mRNA code for amino acids, there's 3-letter blocks, and there's about 20 amino acids in nature that make up all the proteins around us. And as Isaacson implies, we know a lot about the genetic code now, and we know enough to reprogram it to do what we want. And so scientists have even expanded the genetic code, repurposing the letters that used to mean stop to mean things that amino acids that are not found in nature. So giving a protein a little light up tag, or a super reactive chemical group, or even installing sort of modifications that mimic what happens in your cells. And so in these cases, ribosomes aren't doing new chemistry, but they're introducing interesting chemistry via that side chain that we talked about earlier. And CGEM wants to take this one step further. So we don't just want to encode new proteins, but we want to move beyond these alpha amino acids to the routine encoding of even more exotic polymers. So we're taking 2 approaches to this, sort of a bottom-up and a top-down approach. And in the bottom-up approach, it's what I referred to earlier. The new bonds are assembled within the active site of these wild-type and remodeled ribosomes. So ribosomes are literally doing new chemistry. In contrast, in the top-down approach, we're saying, let's let the ribosome do what it's good at. Let's let it make proteins and peptides, and then we'll either use chemistry or biochemistry in order to change the chemistry, the backbone of these peptides. And so turning these circles into sort of diamonds, reflecting new chemistry in the backbone. And in order to accomplish these pretty ambitious goals, we need a few different things. And this is where the diversity of the CGEM team is really critical. So for this bottom-up approach, we need really chemically interesting monomers to incorporate, some of which you can't buy, they're not available off the shelf, and they need to be synthesized by experts. We need to get these monomers onto tRNA so that they can go to the ribosome. We need the whole ribosomal system to be comfortable sort of working with this new chemistry, including all of these accessory translation factors. And for the top-down approach, we can use a lot of this natural translation machinery, but we do need enzymes or reactions that are able to introduce new chemistry. So here I'm showing a cover illustration that we generated with a recent paper. And you can see sort of at the ends of the loops of this protein is a little pentagon right in the middle of the protein chain, and that is not natural. And we were able to install these modifications into the backbone of a protein by looking to enzymes in bacteria that normally make sort of small molecule antibiotics. And a postdoc, Josh Walker, showed that he can get that enzyme to work on folded proteins under the right conditions, and was able to show that introducing weird chemistry like that has real impacts on the sort of properties of this protein. These loops would normally be very wiggly and flexible, but with that modification, it really rigidifies that part of the protein. And I'm happy to talk more about the top-down approach later, but I think it's of more interest to you all and for this audience to focus on the bottom-up approach. So the big player in our bottom-up approach is, of course, the ribosome. It's an RNA machine. It's made of 3 molecules of RNA and over 50 proteins, but these proteins are sort of ancillary. The RNA is what catalyzes the bond forming reaction. It sets up this messenger RNA for decoding by tRNAs, and at CGEM, it's one of our main focuses. So I'm going to tell you a few different stories about different CGEM projects related to the ribosome and how Eterna players have been able to contribute. So first, CGEM began with this mutant ribosome published by some of the team members. So they found this ribosome called P7A7 that if you give cells this amino acid with one extra carbon, it's a little bit longer than it should be. The cells were able to put this into a protein. And that was a really exciting result. That was sort of the first time that we'd seen in a cell, we were able to put this really interesting chemistry into a protein. But when other members of the team took these ribosomes out of cells and tried to look at them and tried to understand how P7A7 works, they found that outside of a cellular environment, the ribosomes don't work very well. And so sort of in these graphs on the left here, I'm showing you just the different populations by size of particles in a ribosome purification. And so little particles like the 30S and 50S are subunits that haven't come together, while 70S is the full ribosome. So you can see in the wild type lots of peaks towards the right, which means there's lots of like fully assembled ribosomes there. But in the P7A7 samples, you can see that there's not a lot of assembled ribosomes. And when we use cryo-electron microscopy to actually look at the particles, we can see that in the case of wild type, the gray part, which is the image that we get from electron microscopy, fits the model, which is the cartoon colors, pretty well. Everything looks like it has sort of a place in the density. But in the case of the P7A7 ribosome down here, you can see that there's a lot of places where we don't see what is supposed to be there. And so particularly sort of this big cluster of blue and yellow over here is where the ribosome active site is, where it's doing its chemistry. And it does not appear to be either folded or present in this mutant ribosome. And if you look at the sort of components of P7A7, you see that it actually hasn't gone through its full assembly process yet. It's missing a few of those accessory proteins, which is an indication that it's not very well folded, not going to be very active. And really it taught us that it's pretty hard to engineer the ribosome, that billions of years of evolution have really converged on something unique and special. And there's a lot of different aspects to consider when we try to make changes, not just the ribosome that we see doing chemistry in its mature state, but sort of all of the things that it needs to do in order to get there. So we shifted gears and we looked a little bit at what chemistry was possible just using regular E. coli ribosomes. And Omer Ad, who was a postdoc in the Shepherd lab, led an effort that looked at a big variety of these monomers. And so he tested them in test tubes to see, what weird chemistry the ribosome could accept. And he found that he could put a lot of unusual things at the start of a peptide. And so you can see each of the different bubbles look really different from each other. And most of them look pretty different from what the ribosome would put in a cell. And he got some hints about what might be possible to put sort of in the middle of proteins and peptides. And another member of the team, Jeff Tharp in Dieter Sol's lab, followed up on this and developed a way that we could put different things in the first position in cells, which required that he engineer the tRNA so that it could interact with things that it normally would not interact with involved in ribosomal incorporation. And so I'm not going to talk about the initiation in vivo work extensively here, but I'm happy to chat more in the Q&A if you have questions. Next, once we knew what kind of things could go in the wild type ribosome, we really wanted to ask how. And so for me as a structural biologist, this is sort of a gut question. I'm more of a, I'll believe it if I can see it type of person when it comes to this biology. And for us, when we can see what's happening, it allows us to think about how we might tweak the system to work a little bit differently. So on the CGEM team, we're fortunate to have Jamie Kate and his lab, who are ribosome structure experts. And they threw everything they had at the E. coli ribosome and were able to solve a structure the highest resolution ever, which means that you can see sort of the most detail that you were ever able to see. And I've included the PDB code here. So I think Slava in the next session will be talking about how to use Chimera. But if you were to fetch the 7K00 structure and download it, you could sort of manipulate it in Chimera and look at all of the different interactions. So this structure showed really incredible detail. So here I'm showing you some of the bases of the large and small subunit RNAs. And you can see a ton of sort of detail. You can see that the sticks, which are the model, fit really well into the electron density maps that we get out of the cryo-EM pictures. You can see holes in the center of rings. That's an indication of like really fine chemical detail in these structures. And you can see that all the sticks fit pretty well in their bubbles, which means that we're pretty confident this is where all of these atoms are, and allows us to make some inferences about what kind of chemistry might be possible. And so we also can see in the structure some interesting modifications to ribosomal proteins, including an isoaspartate residue, which was something that had been observed biochemically 20 years ago. But in all of the ribosome structures, just dozens and dozens of ribosome structures since 1999, no one ever sort of saw that isoaspartate for themselves. It was always just sort of a big blob that couldn't be positioned. And so this is really exciting for us because it means that we can start to ask questions about what types of chemistry the ribosome could accommodate. And this next vignette is really breaking news. And so sort of as a follow-up and a further display of structural prowess, the Kate lab took a step back from the P7A7 ribosome and tried a different strategy for identifying ribosome mutations. So rather than generating a ton of mutants, they worked with the Banfield lab, who studies the genomes of these weird organisms, to identify some archaea that live at high temperatures and have interesting mutations in their peptidyltransferase centers. And so you can see that in E. coli, these bases are a UU, but in these archaea, it's either a CU or a CC. And when they do some biochemistry, they find that at regular body temperature, all the ribosomes perform pretty well. But when you cook the ribosomes first and then ask them to translate peptides, the wild type ribosome loses a lot of activity. And some of these archaeal mutants actually retain a lot more of their activity, which makes sense because they live in really harsh, hot environments. Amos was able to solve the structure, confirmed that the ribosome has all of its appropriate modifications, and was able to sort of look at how this ribosome folds and confirm that it's sufficient and looks capable of doing the same chemistry as the wild type. Interestingly for us, he was able to find sort of a different confirmation of this sugar here, but for sort of general purposes, that's not really relevant. It looks like this is a really good ribosome and a really good starting point for further engineering efforts. And so far I've talked largely about projects that make small or large changes to the E. coli ribosome. But another approach that players have been helping with digs a little bit more into evolutionary history. So as I mentioned, the ribosome today is huge. It's one of the biggest things in the cell, and fully randomizing even 5% of that 23S rRNA requires more atoms than the universe. So we're not going to capture a lot of available mutant ribosomes if we're just sort of blindly mutating things. But ribosomes weren't always so big. And so if you plot sort of complexity or size of the subunits over evolutionary time, you can see that the last universal common ancestor here, way back billions of years ago, had a much smaller ribosome. And so we decided to start from the ground up like nature did. And here you can see the full ribosome. And if I transition here, you can see what Michael Palo designed, which is basically just the ribosome active site, the site that does chemistry in this mini PTC construct. And Michael hypothesized that maybe if you start from something smaller, you could redesign it from the ground up to do more interesting chemistry. So Michael and then Tent tested these using shape and found that the results that players had designed of this mini PTC looked really promising. But a few of them sort of missed the 3D interactions that we see in the full ribosome, where there's a lot of other RNA and protein around. So we're optimistic that the round 2 designs that players generated in the 3D round will perform even better, and Tent will be reporting on those results, I think, kind of soon. So in addition to ribosomes, we also need to obtain charged tRNAs. And so I want to tell one quick story about how players are helping us with that. And so normally when we try to evolve the enzymes that put a monomer onto a tRNA, people look for things like GFP expression or can the protein keep the cells alive? But really in these assays, they're asking 2 questions. They're asking, does the monomer get out of the tRNA? And does the monomer get incorporated by the ribosome? And for us, we really need to be able to decouple those two things because we're working with monomers that might not be very happy with wild type ribosomes. And so lucky for us, nature has the answer here as well in these ribo switches called Tboxes. And so players have been helping us redesign these Tbox switches, which sense whether or not there's a monomer on your tRNA and adjust gene expression accordingly. And so it does this because it's a switch, which is something the players are so good at designing, and we were so excited to get some help with. And so when the Tbox binds to a charged tRNA, this terminator stem forms, and the gene expression is turned off. And normally these are genes related to the synthesis of these amino acids. And so this is telling the cell, I've got enough amino acid around, I don't need to make any more. But when an uncharged tRNA binds to its Tbox ribo switch, this switch shifts into the anti-terminator conformation, and the transcription of these genes continues because the cell is saying, hey, I need some more of this amino acid, let's make some more of it. And we asked players to design new versions of this terminator or anti-terminator switch because in our cases, we found that the difference between on and off was maybe 50%, and we really wanted a broader range so that we could better see activity versus no activity. And so Chintan in the Chatterjee lab asked players to try to optimize this in the Tbox challenge. You all chose, I believe, 12 designs to test, and I believe Chintan is testing them now and should have results to us pretty soon. But it's really exciting to us that players are not only able to help redesign the ribosome, but also to redesign all of these other tools that help CGEM achieve its goals. And so I know I'm running out of time, and so I want to just come back to this slide and say, I hope this really short talk helps you see how players' work fits into CGEM's research program and got you interested in what we might be able to create together. So stay tuned for lab results and more from CGEM. And I'd like to thank the entire team. It's a great group to work with, and I really hope you get to interact with even more of them in the future. And I have a few minutes for questions. William, there are not lab results in this talk, but I hope that Tent or Chintan will be able to present on their results soon at a town hall or something like that.
Can Tent turn on her camera and just wave and say hi? I see that she's here with us. And do you have any, that was great, thanks Sarah. Are there any new challenges on the horizon you want our help with?
I don't have anything exciting to announce here, unfortunately, but I definitely think we'll be returning to the players in the future. And, I think there's plenty of work between sort of where we are and the polymers that we imagine. And so lots of new ribosomes, lots of new, switches and regulatory elements, and lots of opportunities for players to get involved. We're really excited being able to use Eterna 3D and help you all see sort of what we see when we're redesigning the ribosome. And I'm on the developer Slack. If anyone there wants to ask questions or ask for literature, things like that. I'm also in the game, but I don't reply as well to my messages. So I'll put my email in the chat. But if you all ever need sort of direction for literature, then we'll help you out with that. Lorenzo asks, do we ever find designs that results in kinetically trapped misfolded conformations? I'm not sure about that. I would say that the players do a good job of like filtering designs for us. And so if there's a design that would be like obviously not great, we tend not to get those. But I do think that what Tent has found in the mini PTC challenge is that sometimes we get designs that look really close but are not quite folded correctly. And we haven't done sort of a ton of kinetic resolution experiments to figure out how they get stuck there or how they get there. But Tent, if you want to jump in, feel free. Don't feel obligated. Yeah, she's good.
Thanks so much, Sarah. That was great.